So the next speaker is Anna Mitchell, University of Canterbury, Christchurch, New Zealand. The title of her presentation is the Thalamocortical neural circuits, how they work and why they matter in health and disease.
Good morning, everybody, and thank you to Nicola and Haramoshi for organising this and inviting me along.
Do you have slide presentation? Could you just show it, please? Okay.
Just to start off by saying, great talk, Jacob, thank you. And he's obviously introduced the prefrontal cortex very nicely, and lots of the references already, so I'll skip over that, but that was also the intention, because obviously I'm talking about the thalamus. But the thalamus and relationships is what I'm interested in, as Nicola already indicated. Neuroanatomy is all about the relationships. And one of the things in terms of just while they're talking about the slides, sorting out the slides, the tertiary sulci in the prefrontal cortex, but also in other cortical regions as well, they're in the temporal lobes. It's likely also, and people could start looking at this or maybe are already doing so, to be different in people who are neurotypical and to people who have neuropsychiatric, neurological, neurodegenerative diseases. These are tertiary sulci that are associated with different associative areas of the prefrontal cortex, but also parietal, temporal, occipital lobes, and will be differently connected to places in the thalamus, again, in terms of especially the higher order thalamic nuclei that I'll be talking about. And in terms of the links between the columns and then these other representations of neurons, if you look at other people like Ship, Kathy Rocklin, they talk about these signals going up to layer 1 of the cortical regions, cortical mantle, and they're broadcasting information. So they're allowing for connectivity across these regions as well as down within the cortical columns and the lamina, obviously. So there's this ability for the neurons to be able to broadcast and activate these regions and get them back online. And certainly in the work that I've done in terms of the neuroscience side of things with animals, monkeys and rats, we've been able to show that this type of work is what's going on. But I'm not talking about that today. That's a different kind of talk. I'm talking to you about thalamocortical interactions. HDMI cable. If there's a HDMI cable, I can just bring my talk. I've got it on my laptop and just plug it in. There's a HDMI cable, I can just plug that into my laptop.
I think there's a problem then with the recording of the presentation for the YouTube channel.
Oh, okay. I don't mind if I'm not recording.
And there's another point that I wanted to say. I think you rose it. It's the fact that it was Marcello. Paradigms. It's very easy to think of a paradigm to activate the primary visual cortex, right? But it's very difficult to design paradigms that will light up a very specific part of the prefrontal cortex, for example, because it's association cortex. It integrates information from many different sensory modalities, but also from memories, and that is the art of people doing functional imaging. That's where like their expertise comes into play. As far as I'm concerned, it's not just the scans, but designing a paradigm that clearly picks out one single function.
Yes,
Anna,
yeah, definitely.
Stage is yours.
Yeah, but definitely in terms of designing tasks, if you're wanting to engage the prefrontal cortex, it needs to be complex information and across all modalities if you can, but obviously in terms of tracking the different areas of the brain, if you've got visual information, and obviously that would be the most you would have, but also auditory information as well. Okay, so I have no disclosures. Short bio about me. So I got my PhD at Canterbury, where I am back there now, working with rats in behavioural neuroscience. And one of the things that was instilled with me back then was how important it was to be able to understand where the thalamus was and its relationships with the rest of the brain. And from there, I went to a postdoc at the University of Oxford in the UK, switched to monkeys, but also carried on a little bit with rats, and developed, as they got the neuroimaging up on board from 2009 onwards, was instrumental in collecting data in the monkeys. We're still analysing a lot of that data, and some of it is available in terms of Prime D network, and you'll hear more about that as you look across the different models, but I can talk to anybody and send them links later. And then was there till 2002. COVID had an influence on all of that, and thought, okay, I'll head back to New Zealand now. But obviously I also have my family and everything. I'm now an associate professor in behavioural neuroscience back at Canterbury University. So just across the ditch from me to be able to come to this meeting today. So not quite the amount of jet lag that I used to have in terms of other things. So I'm interested in how parts of the brain interact, like I told you, in particular how the thalamus and the cortex interact together. These are called thalamocortical interactions. But we can also think about it in terms of adding the cortex on there again, so cortical-thalamic interactions. My focus on the thalamus is specific to, oopsie, I didn't do anything then. Will it just move on by itself? Okay. I wanted to go back. Is somebody else doing that, or is that me? Oh, okay. All right, wonderful. Thank you. You threw me off a wee bit. My focus in the thalamus is specifically with the dorsal thalamic nuclei and interconnected frontal, cingulate, medial temporal cortex, and these contributions to cognition and navigation. But of course, the subcortical regions are important too. So you've heard a lot already about the prefrontal cortex. You're going to hear some more about the thalamus today, but keep in mind that there's all of these other swaths of subcortical regions as well. So we know a lot from human and monkey research that the prefrontal cortex needs access, and it has access to the relevant information for a given task. It holds this information relevant online during a delay, which we refer to as working memory, but that's just the label of it. And this evidence has obviously been collected from a lot of monkey research where they're doing this electrophysiology recordings. And it organises and monitors the timings of events linked to memories in terms of pulling that information back, putting it in sequences of information, and very, very good at manipulating things. These types of abilities then have been classified as working memory, and there's theories still generating and updating based on further evidence in animal models, especially from experimental findings in non-human primates, but as well as neuroimaging of patients, groups, and neurotypical people. And obviously you've heard all of these references already. These are just a few more from the ones that Jacob's talked about here. The other, Miller and Cohen, for example, was Earl Miller's lab at MIT. But lots of others, and now we can obviously add the University of Miami. And I was part of this Instrument Forum in 23, where we published the book now in 2024. So have a look at that online in terms of the collective, all of these ideas. So if we talk about the prefrontal cortex and its connections then, where are its inputs coming from? So it's got limbic system structures, particularly the medial, dorsal, midline, and anterior thalamic nuclei. Those are the ones that I'm particularly interested in. But it also gets inputs from the hippocampus, from the amygdala. The retrospinal cortex gives it some visuospatial information related to gaze and head direction. You've got your parietal and occipital lobes giving visuospatial information, this dorsal stream of information flow in terms of where pathways, and the what pathways from the inferotemporal cortex and the temporal lobes. So that's the visual ventral stream. These what and where pathways were identified in the monkeys from the work of Morton Mishkin and Leslie Unger-Leider. And then it's got outputs. So again, it's not just this random thing that's working all by itself. It's sending all of this information back to other places as well once it's done something with it. So that information is going to frontal eye fields, pre and supplementary motor cortex, parietal, occipital, temporal lobes, back to the retrospinal cortex, back to the caudate. The caudate doesn't send it information. The caudate, the thalamus, in order for the caudate to interact with the prefrontal cortex, it has to go via the thalamus and the midbrain, brainstem, and cerebellum, obviously. So you can see there, in terms of understanding just the breadth of information that it's dealing with. But I'm not here to talk about the prefrontal cortex. You've already heard a lot about that from Jacob. What I'm particularly interested in is the thalamus. But the thalamus in the womb is contributing to the interactions there already. Those are growing a couple of days beforehand and sending that information up into prefrontal or all cortical regions. So it's working as a partnership in terms of understanding. So when you think about the thalamus, or when you think about any cortical area from now on, hopefully the message from this talk is that you'll also consider its interactions with the thalamus. It's working as a partnership together. So there's no denying that damage to the medial temporal lobes or the cortex means you get deficits. I'm not trying to convince you of anything different. I completely agree with that as well. But damage to the medial diencephalon, especially the thalamus and the mammillary bodies, will also cause deficits. And if anything, they're relatively similar, although there's parallel circuits for this as well. So how could you get damage? You could get damage from strokes or infarcts, obviously. Wernicke-Korsakoff syndrome and dementias have all been identified as having damage in the medial diencephalon. And that's from neuropathological reports. We've known this for over 120 years now from the work of many people who have chopped up their brains and looked at that, and Karl as well. But if you've got patients who are alive and they're willing to go into a scanner, or they can go into a scanner, then we can also see that there's changes in dysfunction in the thalamus and thalamocortical connectivity identified from neuroimaging. So this has then been linked to essentially every kind of neuropsychiatric disease or disorder, illness, schizophrenia, obsessive compulsive disorder, paranoia, the list goes on, dementias, major depression, alcoholism, autism, and pain. So in these disease states, higher order thalamic nuclei are affected more than the first order thalamic nuclei, at least initially in terms of understanding. So these could then be a really good way in terms of understanding some cognitive biomarkers or some thalamic biomarkers or different ways of being able to see the brain in terms of understanding initially, potentially, a diagnosis or a trajectory in terms of understanding along the lines of what a biomarker would do. So what does it mean then? What am I talking about in terms of a first order or higher order thalamic nuclei? So specific thalamic nuclei handle different sensory information. And if you looked on Google or any other AI system, if you picked up a textbook, you would find out that the lateral genicular nuclei is important for vision, medial genicular nuclei is important for hearing, ventroposterior nuclei for touch, pain, and temperature. And what the textbook or any of these things will tell you is that it relays that information. You're getting it from your senses, coming in from the periphery out here in the world. It's being picked up by your senses. It's going directly to the thalamus and then being passed on to these A1, V1, S1 cortical regions. And it's referred to as being a relay. And while that's essentially true on some level, it is adding extra information. Otherwise, what would be the point in it going through the thalamus in the first place? So there's lots of people who are working out what is still going on with all of those kinds of things. Olfactory signals are slightly different, though. They go via the olfactory bulbs just on the other side of your nose and then directly into places like the piriform cortex before they then go to orbital insula and then back into the medial dorsal thalamus. So here in the human brain, you've got two thalamus. You've got one on each side in terms of both different hemispheres. I'm particularly interested. How am I going to be able to show you that? Okay. And the I don't want to push the button. Oh yeah. No. I want to get the pointer. Okay, all right. Well, the one that I'm signaling, the medial dorsal thalamus, but I'm also interested, like you heard, in the midline thalamic nuclei, so that's the intralemma thalamic nuclei, as well as the anterior thalamic nuclei. But you can see, anyway, all of the... it's easier if I can point, but never mind. We'll carry on. So, textbook view. This is definitely the textbook view in terms of trying to understand. They told me that there would be a pointer. Never mind. Okay. So in terms of transthalamic routes of communication, so this is the textbook view, and it's the kind of standard idea. You get input coming through from the periphery here. Let me go on talking. You get inputs coming through from the periphery, going up into the thalamus, these first order thalamic nuclei, going into the sensory cortex, so V1, A1, S1, passing through to sensory motor output, sense and up to the motor areas. In terms of these motor areas represented higher regions, the basal ganglia, superior colliculus, cerebellum, cell groups of the reticular formation of the midbrain and the pons, as well as the spinal cord, obviously. So this sensory information reaches the thalamus in terms of the periphery. It's relayed to the sensory cortex and it's processed through these hierarchical areas, sensory, motor, until it reaches the thalamus. Until it reaches these motor output regions. So there's only this input from those higher order relays. And while that is the kind of textbook view, it's now been updated into this idea of transthalamic routes of communication that are repetitive and ongoing and feed forward and feedback in terms of understanding. This was the work of Ray Guillory from early in the 1990s and has been developed by a brilliant partnership between Murray Sherman and Ray Guillory throughout time. And then Ray sadly has passed away now, but Murray still at in Chicago at the University of Illinois, working, and obviously lots of students and postdocs and people who are now PIs from his lab developing these ideas and carrying them forward. So you've got then your first order nuclei going into the primary cortical regions. But there's inputs coming back from there. These are messages of motor in terms of the blue and also efference copies. So an efference copy means a forward copy of the same information, but that being passed through to these other areas. And that's where there's this idea of this broadcasting of the signal. So information is going directly to these places, but there's also efference copies of information to be able to allow for the streamline of information to be passed to all of these other regions as well. So you've got your direct cortical columns, you've got your direct signaling of information shown by the neuroanatomy, but then you've got these other efference copies of the same information to be able to keep the signal online, represented, keeping things further along in the pathway updated. And this is being maintained and updated and monitored as a consequence of these partnership interactions between the cortex and the thalamus. I'll make sure these slides are all online so people can come and can look back. But obviously in terms of the references, you can go and read more information about all of this. So then these first order relays, then we've got places like the anterior thalamic nuclei, the AD and the AM there, the ventral-posterior nucleus that I told you about, temperature, pain, the lateral geniculate nuclei, the visual processing, and then some of these other ones, the higher order ones that I'm particularly interested in, the medial dorsal thalamus there and the nucleus reunions, and AV thalamic nuclei in particular. And then, as you can see, because there's always a lot of work still to do, and if you're interested and I've piqued your interest, then there's lots of unclassified ones still. So this isn't the same for all of the thalamic nuclei, but it's also to be able to understand that there's differences across all of these. And it may be the case that some of these unclassified ones are also linked to those tertiary sulci and the cortical regions. But of course, some of those tertiary sulci are also linked to these higher order nuclei regions as well. So there's lots of work to do in terms of understanding all of this. The higher order nuclei are larger in volume in terms of understanding that in the thalamus as well. So that's another thing that will work better if you're looking at, voxel sizes and things like that to be able to understand. And then, like as you can see just from here, there's all this unclassified and lots of discussion about what's going on. There's also lots of different classification of this as well. These are relatively new theories, so there's lots of work still that can be done to be able to help classify them better and looking at them across different methods of understanding. So in animal studies, in neuroanatomy, in neuroimaging, to pique your interest in terms of trying to understand. There's this idea of core and matrix output characteristics. So this idea of the core information going to this cortical column, and then the matrix information helping to broadcast that signaling going across in terms of the understanding of these things. So you can see that here for the LGN, it's going directly into layer 4, but then there's the CM that's sending that information in terms of the broadcaster loss across layer 1. So it's this idea of them doing 2 different jobs or even 3 or 4 different jobs as we understand more information. And here for the medial dorsal thalamus, it's going into layers 2, 3 in terms of inputs into those regions, and also information and its outputs are coming from layer 5 and 6. But again, there's this understanding that there's the excitatory inputs in terms of glutamatergic, I beg your pardon, and GABAergic in terms of inhibition. And again, also going to interneurons. So there's all of this in terms of the interactions of these things. So not only where those inputs are coming from, but also where they're landing and the influence they have on those different neurons and interneurons within the cortical layers. So the MD then is a higher order nuclei of the prefrontal cortex. So we've talked about this and set up this proposal. We're working with Murray Sherman and Ray Guillory as a consequence of being able to understand this, setting up this premise to be able to test it and understand, collect more information about it as a consequence. So it's parts of the medial dorsal thalamus that do this, parts of the pulvinar and parts of the anterior thalamus in terms of areas of research that we've been interested in. So why then would the medial dorsal thalamic nuclei be considered a higher order thalamic region? Because it receives inputs from both layers 5 and 6 of the frontal cortex. So there's a reciprocal connection going backwards and forwards. The medial dorsal thalamus goes into layers 2 and 3, but it's receiving inputs from layer 5 and layer 6. All of the thalamus will receive inputs back from layer 6 of the cortex. It's just the way it does. It's helping to do that broadcasting signal. But it's those driver signals coming from the layer 5 motor output regions that is considered to be the signal. And it's an efference copy of those. So this layer 5 provides cortical driver inputs to the thalamus, and there's been identified 2 areas from the frontal cortex to the medial dorsal thalamus. It was the work of Shorts with Goldman Rakesh in 1991 and Helen Barbas's work from Boston University more recently for the magnocellular part of the medial dorsal thalamus. And then Rovo and colleagues with Laszlo Ascardi in Austria have identified mainly cortical inputs to the parvocellular part of the medial dorsal thalamus, these being V-GLUT signaling labeling and others V-GLUT2 subcortical signaling labeling in terms of being able to differentiate out those inputs, while the magnocellular part of the medial dorsal thalamus has a mix of both cortical and subcortical inputs. So it's this combination of all of these interactions together that are helping to understand. And as I indicated, these layer 6 inputs across the brain, they're linked to all corticothalamic projections coming back from the cortex to the thalamus. These are referred to modulators of the thalamus. So they're helping to keep the thalamus online while the cortex is dumping this information to be able to keep the signal going and then passing it back to those other areas of the cortex to be able to help monitor and maintain and keep it upregulated in terms of the attention related to the task that's on hand. And specifically for the prefrontal cortex, obviously associated with things like working memory and maintenance. So what then is the hypothesis in terms of understanding for the medial dorsal thalamus? This is the work that we've been working on for the last essentially 20 years of my career. The medial dorsal thalamus subdivisions enable this linking of online here and now information about what's going on in terms of the current state of the individual. So taking on board what the sensory inputs are coming through. Remember, the medial dorsal thalamus doesn't get any of those sensory inputs themselves. They've all been passed from those primary regions and other areas of the cortex and then passed through to the prefrontal cortex, and then the prefrontal cortex is linking that information back to the thalamus. So it's keeping that information online and allowing those internal states to be able to understand what's going on, as well as keeping on what is the task that I'm doing at the moment. So integrating this information about here I am now, what am I doing at the moment, as well as what's going on in the here and now. And it's coordinating that in terms of motor commands, because those outputs are coming from layer 5 of the prefrontal cortex back to the medial dorsal thalamus to be able to understand, so that the cortex can then benefit from the generation of this coherent stream of choice responses relevant to the task at hand. So the thalamus is telling the prefrontal cortex, okay, I'm engaged now, this task is relevant, it's complex, so dump me some of this information, keep things online, keep them updating for me, so that then let me know what are the choices that you've decided to make a decision about, and I'll pass that information on to these other areas, as well as helping you to keep online, keep the task relevant as we're working in terms of the partnership. So the connectivity between the cortex and the thalamus then, it's glutamatergic, so it's an excitatory driver signal of this information. And this is the case for primates, humans especially, but all mammals obviously in terms of the glutamatergic signaling. And the medial dorsal thalamus is also rich with dopamine signal, noradrenergic signal, or you may also refer to that as norepinephrine. So it's allowing for that attention of the signal, as well as the rewarding mechanisms of the signal, the learning component of that, to be able to signal and help the glutamatergic interactions going together. But we need further evidence. This isn't, as all science, it's never 100%, so we need further evidence, including neuroimaging evidence, and in time, we'll be able to identify more about how these transthalamic communication can explain cognitive deficits associated with many varied neuropsychiatric disorders and conditions. So then in terms of Going a little bit further into the medial dorsal thalamus, like I've said, it doesn't receive any of that sensory information from the periphery itself. It's the partnership with the prefrontal cortex. There's strong reciprocal connections there, and identified way back in the 1940s with Rose and Worsley, saying that historically the prefrontal cortex is defined as the cortical region receiving primary thalamic innervation from the medial dorsal thalamus. So these are working hand in hand. Those connections from the medial dorsal thalamus, as I said already, are established in utero a couple of days before the signals come back from the prefrontal cortex going back into the thalamus. So it's the driver signals going from the thalamus before setting up that thalamic connectivity with the prefrontal cortex that then links the prefrontal cortex back to the thalamus. It's part of the limbic thalamus, so it's considered to be important for cognitive functions, and it's also being referred to as the cognitive thalamus. And in terms of the work that I've been establishing, I've set up this proposition that it's a higher order thalamic nucleus, or at least parts of it, but obviously we're still collecting the data and lots of other people are working on that as well. So in terms of the neuroanatomy and how we can characterise this, so as I'm sure you are all aware, you've heard about the lateral prefrontal cortex already. Different medial dorsal thalamic subdivisions form distinct cortico-thalamocortical circuits. So these are reciprocal connections, so coming from the cortex back to the thalamus, from the thalamus going back to the cortex. And this is in the monkey brain, so differentiating out the dorsal lateral parts and the dorsal medial parts. There's hatching there because both blue parts of the thalamus and yellow parts of the thalamus and orange parts of the thalamus all interconnect to those dorsolateral and dorsal medial parts. Coming round to the sulci there of the area 46, you can see there's a little bit of overlap there, and then the magnocellular part of the medial dorsal thalamus there at the very midline, that blue area, is particularly interconnected to the ventral and ventrolateral parts of the prefrontal cortex. There's inputs then also. The medial dorsal thalamus is in a trisynaptic loop with inputs from the temporal lobes, insular cortex, and also the amygdala. So all of those temporal structures in the insular cortex connect to the prefrontal cortex directly, but they also send a link via the magnocellular part of the medial dorsal thalamus, creating a trisynaptic pathway there, and the work of Helen Barbas has been able to show that especially. And then, as I've already indicated, while we've got all these thalamocortical and cortico-thalamocortical circuits, subcortical inputs are also important. And I referred earlier about the VGLUT2 labelling and the VGLUT1 labelling. So we're, in terms of neurochemicals and neuroimmunohistochemistry, VGLUT2 labelling, if you've got signal in your neurons and it's lighting up as a consequence of labelling and using VGLUT2, then it indicates that it's a subcortical input coming through. And you can see here a nice differentiation. Here, the orange and the green. So the green labelling there is GAD67. It's labelling all the GABAergic cells, connections from the basal ganglia coming through. And then you can see the nice boundary links here of the orange. That's the VGLUT2 labelling, indicating that it's a subcortical input. We don't know where that subcortical input is specifically. The VGLUT2 labelling doesn't allow us to be able to understand that. But we can then go and look at the neuroanatomy and put traces in these regions and work on that. Helen Barbas has already done that, and she's indicated that a lot of this VGLUT2 labelling will be coming from the amygdala pathways that innervate the magnocellular part of the MD. So again, this limbic cognitive functioning, so it's getting inputs from the amygdala associated with reward, fear conditioning, all of that. It's all been shown in rats and monkeys and in humans as well. We now know, and again, in terms of the lighting up of those signals, depression, anxiety, post-traumatic stress disorder, all of these things are changing as a consequence in the medial dorsal thalamus and those magnocellular thalamocortical connectivity regions. And then, of course, I work with monkeys and with rats and humans. There's comparative rat and monkey PFC MD connections. So this is across all mammalian species in terms of trying to understand. Obviously, it's not as complex as a human or a monkey in terms of the rat brain. It doesn't have all that lovely gyrus and sulci. In terms of seeing here on the rat brain there, this second column there, you can see it's all smooth. The rat brain is smooth. It doesn't have any of the cortical folds like the monkey brain and the human brain does. But in terms of grandness of scale and understanding, each of the species is relevant for different neuroanatomical questions, neuroimaging questions, and cognitive and behavioural neuroscience questions as well. And of course also molecular. So then in terms of damage to the MD, different pathways showing different understanding and MD-PFC interactions resulting in cognitive deficits. So in humans, thalamic stroke, so the work of some French researchers, Danette, and others looked at thalamic strokes. So again, developing neuroimaging to be able to quantify where these regions are in the brain and understanding what's going on. Schizophrenia, the work of many people around the world identifying now different parts of the thalamus, especially the medial dorsal thalamus, the pulvinar, particularly in terms of schizophrenia. Wernicke's-Korsakoff syndrome, again, some French researchers, especially Anneliese Pitel and others there. Frontotemporal dementia with Bochia and others at London universities. They've all spread out now in terms of looking, but again, the references you can see there, and they've been using neuroimaging to be able to identify these changes in people who are obviously still alive. We'll wait for the neuropathology reports to come in time, obviously. But then also in affective disorders, anxiety, depression, lots of recent reviews on this, and then more recently in paranoia as well. And then, like I told you, we've known for a long time that alcohol affects all regions of the brain. Wernicke's-Korsakoff syndrome especially affects places in the medial diencephalon, including the medial dorsal thalamus, the mammillary bodies. So work here, an Australian group in Harding from 2000 in brain, identifying these areas of the brain that are changed as a consequence and controls Wernicke's-Korsakoff syndrome and Korsakoff's psychoses, to be able to understand the distinctions. And then the work of Anneliese Pitel and colleagues there showed damage in the medial diencephalon and alcohol use disorder, Wernicke's encephalitis, and Korsakoff syndrome. And you can see the thalamus, medial dorsal thalamus, anterior thalamus, and the mammillary bodies are all significantly damaged as a consequence of developing not only just alcohol use disorder, but with combination with Wernicke's encephalitis and Korsakoff syndrome. In non-human primates, and of course it's different talks altogether, but just highlighting some of the differences. I'm running out of time, so I don't want to just read off the slide, but you can see the medial dorsal thalamus or medial dorsal orbital frontal cortex, ventrolateral prefrontal cortex interactions are linked to cognitive deficits, and obviously in rodents as well, goal-directed behavior, cognitive flexibility. In monkeys, we can look at forward planning, adding the temporal component to things as well, reward evaluation, visuospatial discrimination learning. So keeping all of these things online, the complexity of the task really makes a difference. And more recently, some of the work we've been doing has identified paranoia as being also a key thing that's linked to the medial dorsal thalamus in monkeys. So then to finish, higher cognitive functioning. Like I told you already, the hypothesis that we have is this idea that the thalamic structures enable the linking of online here and now information. Some people would refer to that as being working memory, but it's not just working memory. There's a link of working memory, but also attentional components, as well as what's going on in terms of your internal state. And it's linking these things to the motor commands that you're doing. And it may not be that you're even making a motor command. It may be some intrinsic decision making to not make a motor command, but rather just to have those thoughts being generated. So in this case then, if the thalamus and the cortex are interacting together, the thalamus will then benefit from this ongoing updating, as people refer to it. But it's also really, in terms of more scientific, modification and maintenance of information. So we can have a coherent stream of choice responses that are relevant to the task in terms of keeping the cortex updated about what's going on. How does it do this? Again, the jury's out in terms of trying to understand. In terms of, we talk about signal as well, in terms of increasing the signal. We talk about reducing the noise. We talk about enhancing coherence and synchrony and the ability for different signals. In electrophysiology, we look at the frequency of the wavelength, but also when the spikes are happening at different places along the wavelength, if it's happening in the peak or in the trough or on the upward slope or in the downward slope. So it's all this understanding that we have in terms of the electrophysiology of the work that I do. The connectivity between the cortex and the thalamus is glutamatergic, as I've told you, and in primates, especially the medial dorsal thalamus, is rich in dopamine, noradrenaline, and norepinephrine. So there's components of attention and reward, as well as that linking with the working memory. It's essential in terms of future behavioural, cognitive, and molecular neuroscience research that we combine methods, including neuroimaging, genetics, discrete brain manipulations, drugs, chemogenetics, optogenetics, the whole raft of understanding, so that we can understand more about these thalamocortical interactions as we're investigating them over time. And it's likely that the thalamus especially will provide effective treatment options for future studies in terms of understanding how we can manipulate the brain so we can help find ways to be able to improve the quality of life for people with neurological conditions and disorders. It's really difficult to be able to treat all of the prefrontal cortex, but in terms of understanding that the medial dorsal thalamus in a human is only a few millimeters wide, and we can be more selective with the receptors that we target or the specific kinds of neurotransmitters. Yet it's broadcasting to all of the prefrontal cortex as well as these selective columns. If we can understand how it's doing that in these ways, then it seems like a more worthy target, or at least that's the understanding that we have at this stage. So where to next? If I've picked your image, the thalamus and neuroimaging, I recommend, this is a recent review, Neuroscience Reviews of Nature of Reviews of Neuroscience, that you start looking at some of the work of Zilles and others, obviously some of the people that I've referenced here in terms of understanding Emmanuel and Lise Michael Hornberger, to be able to understand how you can learn more about finding out about the thalamus and including it in your neuroimaging studies. Thank you very much for your attention, and I look forward to seeing more of your work to be able to understand what it is that humans are doing when they're alive and involved in cognitive tasks, not only with the thalamus, not only with the cortex, but please, please, please look at the thalamus as well. Thank you.
Thank you very much. So we have time for a couple of questions. So if you have any question, please just come to the microphone. Yes, please.
Thank you so much. Very interesting. So you showed a slide with the Wernicke's-Korsakoff syndrome where I believe the locus coeruleus was 100% affected. And I'm wondering if you could speak a little bit to, in that disorder and in dementias, how the pathology might be coming perhaps from the brainstem, possibly locus coeruleus, and what those pathways are.
Yes, exactly. So the locus coeruleus is the noradrenergic or norepinephrine. Some people, it's essentially the same, not exactly the same, but noradrenergic pathway. And it's mainly projecting to the hippocampus and the prefrontal cortex, but it also projects via the midline thalamic nuclei. And that's where the key is then again in terms of interacting with these regions. So the medial dorsal thalamus receives noradrenergic input from the locus coeruleus, and the prefrontal cortex does as well. So then it's signaling the same thing going into those two different structures that are then also working in a partnership together. It also sends inputs into the anterior thalamic nuclei and the nucleus reunions. Those are the two midline thalamic nuclei, and to the hippocampus. The anterior thalamic nuclei and the nucleus reunions connect to the hippocampus and also to the prefrontal cortex. So it's sending that signaling to those two key subcortical regions, although you could call the hippocampus a cortical region. And then the hippocampus is then sending inputs to the prefrontal cortex. But the prefrontal cortex doesn't send inputs back to the hippocampus. It does that via the anterior thalamic nuclei and via the medial dorsal thalamic nuclei and via the anterior thalamic nuclei. So there's these wider circuits in terms of understanding all of these regions, and you need to be keeping those in mind when you're looking at your individual particular regions, for example, the hippocampus and the prefrontal cortex. You need to be considering these thalamic inputs, interactions with those, as well as the brainstem inputs as well. So yes, the neurogenergic system, the locus coeruleus, is pretty much wiped out in people who have Wernicke's-Korsakoff syndrome. Wernicke's encephalitis, and Korsakoff syndrome.
Thanks for your talk. I had a very naive neuroanatomical question.
There's no such thing. We're all learning. Nobody knows everything.
That's true. I'm just curious, are there connections from the thalamus to all parts of cortex, or is it just...
Absolutely.
So, if you look at any cortical region, there's probably a connection to thalamus?
There's absolutely a connection to thalamus. The thalamus, like I've told you, is started in it's a subcortical region. It's sending the projections to all cortex in the womb before the cortex is sending the projections back to the thalamus and to other areas like the basal ganglia as well, setting up the hippocampus, obviously, in terms of understanding. But yes, all cortical areas have a relationship with the thalamus, just different parts of the thalamus, different nuclei within the thalamus.
And are efferent and afferent projections just kind of intermixed next to each other, or are they kind of organized by nuclei in the thalamus?
They're organized by different subdivisions. in the thalamus, so nuclei within the thalamus, but then subdivisions of the thalamus. And as you saw, then in terms of the layers, those projections will go to different layers of the cortex and come back from different layers of the cortex. So, for example, LGN will send, or all sensory inputs from the thalamus go to V4. Sorry, they don't. They go to layer 4 of the V1, S1, A1. Okay, because that's the driver signal into that layer 4. But they also send projections to layer 1, and inputs come back from layer 5 and layer 6. And there's recurrent feedback and also feed-forward loops. So there's this ongoing loop of information coming through. And they're using different cortical layers to be able to help with that signaling and communication.
So, any other question? Yes, please.
No, I just want to borrow the microphone.
Okay. Sorry. So, anyone else? Okay. Thank you again very much for Anna for a wonderful talk. Before moving to our next talk, we will have a very short break. Then maybe the next session will start 10:30. Is that okay? Yep. So very short break. Yes.